Welcome to this webinar. We are today covering the topic, protecting our natural assets. We are doing that through nature conservation and responses to climate change. We have 3 guests with us today, so you can look forward to interesting talks from them. Before we get started, maybe we can just introduce the NSCF, or Natural Science Collections Facility, for those who are not familiar with us. We are a network of institutions all over South Africa that hold natural science collections. By natural science collections, we mean dead plants and animals that are preserved. So they are preserved for research purposes, and they are also considered our natural assets, hence the title of the webinar for today. So would like to welcome you. My name is Furupero Tambani. I am the Science Communication Officer for the NSCF, and I am with my colleague behind the scenes, Chanel Ribeiro, who's running the slides and helping with the technical aspect of this webinar. You can find us on our social media platforms, on Facebook, Twitter, Instagram, on our YouTube channel, as well as on our website, nscf.org.za. And we are also on LinkedIn. It's just that we are very young there, and we've also recently opened a TikTok account, so we're also very young there. But you are welcome to interact with us there. Before we get started, we just need to get through some guidelines for engagement, just so that everyone knows how to behave in this space. So we ask that you keep your contributions as helpful as possible and considerate of the hosts as well as other participants. You can use the chat box below. So if you go to chat, you can say hello and let us know which organization you're coming from. Thank you, Ian. Ian got us started. Ian is from the NSCF Hub, so he's part of our team. So you can join Ian in greeting everyone in the room. Just let us know where you're coming from, which organization, which school or university, or whatever is applicable to you. This is just African culture. It's in us, it's in our DNA to get into a space and greet everyone that is there. So let's get that going as we get started with the webinar. Continue doing that as I speak. I see, let's see here. I see Robin Lyle got started also in greeting. She's one of the guests today. We have Renee, Rihanna, we have Petro, Mary. It's good to see everyone. Nikisha also, who's also a guest today. So let's get greeting as I speak. You can also use the Q&A box to add your questions there. You'll find that as we speak, as we go through the webinar, there might be questions that come up to you, just so that you don't lose, we don't lose those questions, or they get lost in the chat if you go to the chat. So please don't use the chat for questions. You can just use the Q&A box for the questions, just so that we don't lose them. And if we don't get to answering your questions, we will try to get them answered on the webinar link. By this, we mean that when we are done with the webinar and it's been edited, we're going to upload it to YouTube, and we can get your questions answered there. Another alternative is our Facebook page, because we are live on Facebook now. You can also use Facebook to ask your questions, and we'll get them answered. So please be patient with us if we don't get to answering your question. We will do that. Please don't forget to visit our website, It's www.nscf.org.za for any information on natural science collections. Other people know them as natural history collections. Either way, if you want any information regarding natural science collections, you can get to our website. We have loads of information there. These webinars are linked to Showcases, a publication that we have started. We have been doing this over the past year, and this is the first one for this year, but we already have 4 issues. So today we'll get started with Nikisha Singh. She took over as the curator of the Skukuza Biological Reference Collection in December 2020. And the reference collection serves as a valuable resource to researchers and anyone that is interested in the flora and fauna of the Kruger National Park. Yes, you heard that right. She works in the Kruger National Park. She loves her job because it entails various aspects. At times, it may be working on identifications of different species, going on collection field trips, and experiencing the different and beautiful landscapes of the Kruger National Park. And she also likes assisting researchers and colleagues, and a whole lot more. As you can imagine, if you work in the Kruger, the Great Kruger National Park, obviously there's a whole lot more to explore and to work on. So Nikisha is going to speak to us today on preserving the past for a fruitful future. Nikisha?
Thank you so much for the introduction, Fulu, and a very good morning to everyone. I'm going to start by first giving an overview of the importance of museums and herbaria, and then I will. So what type of specimens can be found in natural science or natural history museums? It might be an entire animal skeleton. At other times, it might be entire animal specimens that have been preserved with skin, fur, or certain characteristics by a process known as taxidermy. So at other times, it might be just a part of an animal specimen, such as the skull, teeth, or preserved skin. Many of us know what museums are, but what is a herbarium? Okay, so herbariums are basically collections of preserved plant specimens. Plants are collected in the field for various reasons. It might be for interest or for research purposes. Once these plants are collected, they are then dried, pressed, and mounted onto herbarium sheets. The sheets also contain labels with important information that is collected along with the specimen. And we will go into detail about what kind of information needs to be collected a bit later. So why are museums and herbaria important? Okay. For research purposes, there are new and interesting discoveries being made every day. Researchers sometimes need to refer to the past species, specimens, in order to find out where a new specimen or new species needs to fit in. Taxonomy deals with the classification of animals. Name changes happen all the time. Therefore, it is always important to have an up-to-date record of name changes and such to refer back to. Studies dealing with the structure or certain characteristics of animals can also be done by using specimens that are stored in a museum or herbarium. Okay. It is also a place of education. If you are interested in a certain animal or plant, you can find out so much of information by visiting a museum or herbarium and speaking to the curator in charge. It is also a place to store specimens safely for long periods of time under the correct conditions. Okay. Museums and herbaria also act as an inventory. For example, a museum in a specific city or province will have a record and collection of plants and animals specifically collected within that area. We now know that museums and herbaria play an important role in research and contain a wealth of knowledge. But as the world has now progressed, new types of technology are coming out all the time. If you need to find out something, all you need to do is click a button by using Google or even an app on your phone. People no longer find the need to visit museums, herbaria, or places that offer a wide range of knowledge. But specimens that have been physically collected hold a wealth of information and cannot be replaced just by pictures. Physically collected specimens are important because they have certain characteristics that can only be seen on the physical specimen. For example, fruit that have been collected along with plant specimens. One can feel the texture and even get an aroma from them. DNA studies are also important, and we need physical specimens in order to extract that DNA. Okay. So there is certain information that must be collected along with any specimen in order for it to hold more value. Okay, so where the specimen was found, this would include major locality and minor locality information. So your minor locality would be, for example, a city. Major locality would be province. GPS coordinates are also important. So if a researcher or any person wanted to go back and find the specimen, they would have exact locality points. Scientific name and common name. It is important that these specimens are ID'd by experts and that correct scientific names are given. The date the specimen was collected, who collected the specimen, any interesting or important information. For example, if you were doing plant collections and it was flowering, then it would be important to note down the flowering time. Information on habitat. Was the plant or animal collected near a river or a mountainous area? And information about certain characteristics that might be lost when the specimen is preserved. For example, flower color or aroma. Now I'll give just an overview of the Skukuza Biological Reference Collection, which I oversee. Okay. The first formal plant collections were done in the 1950s. Okay. And animals collections were started around 1959 up until 1970. We now have more than 14,600 animal specimens, which are broken down as follows. 10,000 insect specimens. Some of these are pinned specimens, while others are preserved in 70% ethanol. Our reptile, amphibians, and small mammals are also preserved in 70% ethanol. We have bird study skins, and the invertebrates are preserved in 70% ethanol. We also have approximately 16,000 pressed plant specimens. Why is this collection in particular so important? Okay, we act as an identification service to Kruger National Park staff, visiting researchers, or any member of the public that might be interested in knowing about the flora and fauna of the Kruger National Park. We are building up a database or inventory of what plants and animals occur within the park. By having these records and locality records, we can determine species distribution trends and population trends. So if a species might be decreasing or increasing, or if there are certain management or conservation decisions that need to be made. A lot of research and studies happen in the Kruger National Park. Researchers also visit the museum and herbarium to have a look at specimens. And one of the most important is the educational purposes. We do host talks and workshops for staff, schools, visiting colleges, and any member of the public that might be interested. The way forward, we would like to inform and educate people on relevant topics and research in nature and conservation so that future generations can aid in conservation efforts. Okay, thank you.
Thank you, Nikisha. And
Thank you so much.
Thank you. It's interesting to see how the Kruger National Park is not just about the animals and looking at them, watching them. There's also some background work that's being done, some research being done.
Yes,
And I'm so glad that it's being done in a museum, in a natural science museum or herbarium. So thank you so much for letting us know that. Right. We will then move on to Dr. Lizo Hugo Coutier. She is from the National Museum in Bloemfontein. Lizo is the principal museum scientist at the National Museum. She's also the head of Department of Terrestrial Invertebrates there. And her research focus is the taxonomy and ecology of soil mites, also scientifically known as Oribatida. And she has been doing this for the past 15 years. She loves to work in the mite collection and to discover new mite species from this collection. I'm interested to see what mites have to do with climate change and nature conservation. So, Lizo?
Thank you, Fulu, and hello, everyone. My presentation is about a recent study that was published in 2021. I will also link this to the Acarology collection of the National Museum. So, as you know, people living in different areas of the world are adapted or are used to different temperatures. I think, for example, Eskimos will have a very hard time living in the Sahara desert. Even in South Africa, if you grew up in the cold West Coast, it is quite uncomfortable to go to the Natal coast in summer for summer holidays with the high temperatures and humidity. So simply put, in a recent study, it was found that intertidal mites also have temperature preferences. Let's break it down a bit. The intertidal zone at the beach is the area of the shore that is submerged at high tide and exposed at low tide. Low tide is when the ocean water reaches the lowest point on the shore, and the high tide when the water reaches the highest point. Maybe you can remember when you went for holiday as a child to the beach and you were like exploring the rock pools and seeing small fish and starfish, and that is the intertidal zone. So as can imagine, this zone can be physically stressful for the animals living in it because of dehydration risk, temperature extremes, light intensity, and salinity fluctuations. But these mites are specially adapted for submersion in water at high tide, although they will not be able to survive in open water. So some mites, as adapted by feeding and moving, it's synchronized with the tides, and they can become less active when submerged. And they might also escape to above the water line during the high tide. And mites can crawl around, climb around. They creep into the crevices in the rocks and barnacles and algae, and they hook fast with their strong hooked claws so that they're not being washed away. Just for those that's not familiar with mites or Oribatic mites, Oribatic mites are also known as moss mites or beetle mites or soil mites. They have a hard body They have 4 legs and 1 pair of sensory organs, and they are between 0.3 and 0.7 millimeter in size. So worldwide, there are more than 11,000 species known. In South Africa, there's about 500 species, but worldwide, there's only 80 species known that are marine associated. So in general, Oribatic mites are detritus feeders, and for marine mites, they feed on algae, decaying plants, fungi, and seaweed. There are 4 species of marine mites along the South African coast. 4 species of 3 families. Here are the pictures of them. So the families are Purocaridae, Fortunidae, and there's 2 species of Oribatic. From the many global studies about intertidal Oribatic mites, it is clear that the Purocaridae mainly occur in the southern cold temperature regions and southern polar regions, while the Fortunidae and Oribatic are found on the subtropical and tropical shores. Globally, there are no overlaps of the cold and warm adapted species, but on the South African coast, although there is no overlap, the cold adapted and warm adapted species occur very close together. So all these species were collected during fieldwork in 2019 and 2020. We collected at approximately 16 beaches, evenly spread along the South African coast. And luckily, we also included Umhloti and Umkomaas in the KwaZulu-Natal, which was actually recently hit by the floods. This study was a collaboration between South Africa and Austria. So the coastal climate of South Africa is strongly influenced by the cold Benguela current on the west coast and the warm Agulhas current on the east coast. Due to the influence of these currents on the coastal climate, the South African coast has been divided into 4 ecoregions close to the coast, namely the southern Benguela ecoregion on the cold west coast, the Agulhas ecoregion, which is the warmer south coast, the Natal ecoregion, the warm subtropical east coast, and the Delagoa region. It's the tropical east coast from St. Lucia upwards towards Mozambique. The terrestrial climate has also been divided into regions, but the coastal ecoregions and the associated sea temperatures explain the geographic distribution of the mites better than the terrestrial regions. Just to explain this better, so P is for Purocaridae, which is the cold adapted family. S is for Oribatic, and F is for Fortunidae. They are the warm adapted families. So as you can see in this picture, that the study found that intertidal mites were absent from the southern Benguela ecoregion. That's the cold west coast. The Purocaridae occurred in the Agulhas ecoregion. It was from Komiki along the southern coast up to East London. And then the Oribatic, one of the species, was found from south of East London up to Durban, Natal ecoregion, and the Fortunidae from Port St. John's to Durban, also in the Natal ecoregion. And there was 1 species of Oribatic, Oribatis divergens, was the only species sample in the Delagoa region at St. Lucia. So one might ask why the mites of the different families are not able to cross the temperature borders. Well, maybe as explained in the first slide, mites are not able to survive outside the critical temperature ranges they need to for reproduction and development. Here is another graph indicating the distribution of the different families superimposed on the currents along the South African coast. The distribution confirms to the global trend of the cold adapted and warm adapted families. Just for interest, we did sample along the west coast up until it was close to Honderklip Bay, but we didn't find any Oribatic mites there. So the climate of South Africa is changing, with certain regions showing significant warming and rising sea levels. So how will these changes affect the intertidal mites, which are so dependent on temperatures and habitat availability? So increasing temperature may alter the geographic occurrences of mites. For example, the warm-adapted families may expand their ranges southwards, and the cold-adapted Purocaridae family, their populations can be reduced to a few southwestern coastal areas. Increasing sea levels will not have a very large effect on mites, as they can easily move to higher areas, except in cases where rocky shores are bounded by sandy beaches, resulting in loss of habitat and extinction of those local populations. So how does this study relate to the collection? Just a bit of background, the archaeology collection of the National Museum consists of more than 270,000 specimens of Oribatic mites from more than 4,700 localities. However, before this study of 2021, there was only 180 specimens from Halossetus capensis, which is the Purocaridae family, deposited in the collection. And despite there were earlier reports in the 2000s of the other species. So the importance of depositing specimens in museums' collections was recognized in this study, because comparisons of the new specimens could not be made with the specimens from 20 years ago, because we couldn't find them. But there's a bit of good news. After the conclusion of this study, we actually found some of the specimens, including holotypes from the early 2000 studies. It was discovered somewhere in a box under someone's table in Europe, and it was donated to the museum. Unfortunately, some of that specimens dried out, and some of it was lost. Even a holotype was lost. But luckily, we could designate a new type for that species. So now we have material from this recent study and material from the previous studies that was donated to the museum. Just an example, a previous study mentioned the species, but just as it was like Schusteria species 2. So we couldn't find that specimen to confirm which species it is. So we will never exactly know, but we think it will be Selenuribatus divergens. So luckily now we have more specimens, and this can now serve as a baseline for future climate change studies. Thank you. That's my story.
Thank you, Lizelle. What an interesting talk. So the immediate thing that came to my mind is that if we don't have species that are preserved in the museums that were endemic to KZN, those areas that were hit by floods, then that would mean that we have no record of them, which is such scary news. And I'm just hoping that we don't have any species that were lost that we'll never know about. Just to show the importance of these collections. So thanks so much, Lizelle. Up next, we are moving to Robin Lyle. Robin is not new to this platform. We've heard her before. She was talking to us previously about the fall armyworm, but today she's talking to us about something different. Robin Lyle is a senior researcher technician whose first love is the spider, but recently she has been asked to assist with training activities within the ARC. She had done extensive work on the infamous fall armyworm, which she has presented on this platform before, like I mentioned. She has been doing taxonomy since her honors year and has helped out with training activities for the last few years in the ARC. Today, she'll be talking to us or telling us to protect the spiders in our backyards. She's going to explain why. Robin?
Hello, everyone. Thank you for the nice introduction, Kulu. I'm a bit of a jack of all trades, but my first love is spiders. The work I'm going to talk to you about today is actually a very long-term process. It's been in plans for years, actually, and I feel I can't take credit for it. It's someone else's long-term dream that we're going to share, but I'll get to that in a little bit. So basically, what I'm going to talk about today is the redlisting of the South African spiders, and none of it would have been possible without the National Collection of Barracuda. The collection was established in 1976, and it forms part of the National Public Goods Assets, and it houses different arachnid orders, but the largest order is that of the spiders. There are over 300,000 specimens of about 80,000 repositories. So it's quite a big collection. It's one of the largest in the country, and we have a great team who looks after it and a very good collection manager. So the work we're going to present today actually started with Dr. Anthony de Vries, and she was the founder of the National Collection of Arachnids, and she had an amazing career. She's very, very prosperous woman, dynamic, very energetic, and if it wasn't for her, I don't know if African arachnology would be where it is today. Just some of the highlights from her career is she did the African Spider book, which was one of the first identification manuals for South African spiders. You can still get a few copies. I think that we have like 4 left in print. It's very outdated, but it's a very good book. It's what I started to learn my field on. She published numerous papers, and she was a key player in the South African National Survey of Arachnids, also known as SANSA. And this work that she did at this project is actually where we got, how we were able to came to the point where we could redlist the spiders for the work that we did. And like I said before, she was the founder of the collection, so she's worked here for more than 46 years, and she has retired for quite a while now, but she's still working on spiders every day. I don't think that will ever change. So just to give you background, the South African National Survey of Arachnids, or SANSA, had a number of aims, and when it was started in 1997, these aims were the work that the aims we're going to present seem very, very elementary, but actually the work that without these simple aims, which aren't that simple to execute, the work wouldn't be possible. So the idea was to discover, describe, and to make an inventory of all the South African spiders in the country, to get all the information into a database, then to synthesize and analyze the information, and then to make the data available through science to the society and the public, to use the information for sustainable use and conservation planning, and to develop products that meet the need of the community, and to make people aware through education and training, and to help build capacity and infrastructure. So this is the aim of the project, and I'm just going to briefly discuss how they went about executing it. Like I said, the project was from the 90s until now. So it's not a small project. It was a long-term project that took a long time to get to where we are now, or where the collection is now. So when the project started, they built a relational database that contains information from all the specimens in the collection. They consolidated all the data, and they included data that was found in publications. That's why you know that specimen was actually accurately identified. And they did a gap analysis. So they worked out where in the country have we sampled, where we haven't sampled, and then we went about doing field work in the gaps. And there's still massive gaps in our country that need to be sampled, but every specimen collected helps to contribute to filling those gaps. And then once you've got all the specimens, obviously you need to identify them. And so did a lot of work towards that. When I started joining, we started running awareness or sorting workshops with the Spider Club of South Africa. There's training, which it could be training of interns or students, informal training. We also do education, where we give talks to the public on what a spider does. It can be to small children or to adults. It depends on where the need arises. Recently, I just gave a talk to the poor trackers on one of their camps, where we actually went out and helped collect some spiders with them, which is very entertaining. It's fun because it's not something normal most people would ever do, is go out and collect a spider. Then product development. Auntie is very well known for all her books that she produced, and they've really contributed in numerous ways. We have some for sale. They're very helpful. They're obviously, with taxonomic changes, they're not all up to date, but they're still very, very handy. And she's busy working on the second edition of another field guide, which is very nice. But one of the most important things that came out of it was the conservation assessments and the red listing. So what is the conservation assessments? So what they did is they worked out the level of endemicity for each species of spiders that were known. And you can see it goes from 0, it's a cosmopolitan distribution, 1 is afrotropical, and it breaks down up until the higher values where it's literally known, number 6 is where it's only known from one locality or the type locality. And then local, a value of local abundance. So it's rare, it's only known for 1, 2, 3 localities. 3, rare if it's known from 4 to 10 localities. And then a 1, it means it's abundant, it's known quite well. And you can see there's a breakdown of one of the jumping spiders of how it would work. This helped to give us a taxonomic status, if it's very endemic, is it rare, is it cosmopolitan, which helped us decide which species needed to be assessed. So how did we get through the red list assessments? Obviously, we did not do this by ourselves. Red listing is a very complex, well, for me, this is not my everyday job, complex process. We were approached by the SAMBI Threatened Species Program, and they help red list or evaluate each species on the South African spiders on the IUCN red list through the categories and criteria that they use. Obviously, those that have very wide distributions, most of them came at least concern. There's a number of data deficient ones. But what we found is that there are 3% that are considered threatened, 32% are data deficient, which means there's a lot of work that still needs to happen. In those cases, often it's only known from a male or a female from a type locality, or there aren't very good scientific drawings to go with it. 3 are near threatened or rare. We have a large number of rare species that are endemic only to South Africa. There's even some of our families that are only endemic to South Africa, which is very cool. And the bulk of them are least concerns, which means they don't have considerable threats, they're widely distributed. And for the red listing process, we included everything up until all taxonomic changes up until 2020. You had to draw a line at some point, otherwise we keep on going on with it. So we were very lucky because of this work, which is a long time coming. We have a number of publications as well as conference contributions that have been presented nationally and internationally. As you can see, the red list poster that we have, and a number of popular articles that have been written by it, and case in point, the current talk we're giving. And then how was this information used? So this part for me is actually very, very cool. It's not something that I've used myself, but it's so nice to see how the data from a specimen has gone all the way through the entire process and is now actually being used in the real world. So what happened is this red listed spiders for this country have been included in the Species Environmental Assessment Guideline. And if that guideline is then linked towards the national screening tool that was developed by the Department of Forestry, Fishery and the Environment. And when you do an environmental impact assessment, you actually have to check what species are in area, and you plop in your information. I'm not the EIS specialist, so I'm not sure exactly how to explain the process, but you use the screening tool to check if you could possibly find that spider in your area, and it helps you, it forms a form of protection for those invertebrates, for our spiders. So as much as we have our big endangered elephants, we also have our endangered spiders being protected, which I think is very, very cool. So where are we at now? We're busy helping SAMBI capture the data on IUCN database so that these assessments can be recognized internationally. SAMBI's got staff working on it, and we have some staff who are coming on board to help work with it. And the red list process is revised every 5 years. Obviously, we don't have to redo all of them. Then it would include any taxonomic changes and new species that have been described. So it's quite, I mean, SAMBI is very active in these processes. They've done it for a lot of the plants and a lot of the butterflies. And then in conclusion, it's really amazing how from one specimen, or from the specimen data, not even the specimen itself, the data associated with the specimen, this process was possible. And as I said, the collection was started in 1976, and it's been going slowly. With Auntie's vision, we've worked towards the point where we are now. And it is really a team effort with SAMBI, a team effort with colleagues and collaborators within South Africa and internationally, the people who helped assess the species. So it's really been a very, very insightful and informative process and quite a bit of work, but it's very cool to see how it actually is being implemented on a national level through policy that's in legislation in our country. So I must say I'm very excited about it. Thanks. Thank you, everyone.
Two things, right? So I just love how you lit up when you spoke about the screening tool. It's like your face it just lit up. The second thing is I just appreciate how you are giving, this term of giving flowers. You're giving flowers to Ansi while she's still here. So thank you for that. Thank you for such an interesting talk. So insightful, and it is so exciting to see the work that came from a specimen having results or having a tool that people are using now. That's so amazing. So thank you for sharing that. That was the last of our talks. Thanks everyone. Thank you to all our speakers. We will now go into the question and answer slot. If you have any questions that are coming up for you now, please do put them in the Q&A box so that we get them answered. But the first one was from Rihanna. She was addressing it to Nikisha. So Nikisha, are there any mushroom or fungal specimens in your collection?
There are no fungal or mushroom specimens in the collection at the moment, but we are working on a project that will bring in that type of collections. So that project is in the process of being, going through some procedures and stuff. And once all of that has been finalized, then that collections will also start.
All right, thank you. I'm sure Rihanna is so passionate about fungi, so that's why she's asking. I'm sure she's thinking of doing some work with you guys.
We definitely will be having specimens coming in here. We're just waiting for that project to be sort of, with all the guidelines and stuff. Like, we're just following all of that. And once we're in the final steps, then the field work will begin and collections of fungi and stuff will start.
Awesome. Another question for you, Nikisha, is from Sfiso Ngati. I'd like to know if there are any species that are endemic to Kruger National Park which have a specimen that's housed in the Skukuza reference herbarium.
Okay.
Did you get that?
Yes. Okay. Specimens that are specifically endemic to Kruger National Park, I'm not 100% sure, but I am sure that species that are probably endemic maybe to I'll have to find out more information on that and just check our records, and then I definitely will get back to him if that's okay. Yeah. So I'm not sure if there is like a specific endemic species in Kruger itself. Yeah.
Okay. All right. Thank you so much. That's all the questions we have in the Q&A box. Thank you so much for attending this webinar. It was quite interesting, and you can keep in touch with us on our social media. So if you are on Facebook, we are at NSCFSA, on Twitter, NSCF_SA, Instagram, NSCF_SA, on YouTube, which you should definitely go there, because the recording of this webinar is going to be uploaded there. It's Natural Science Collections Facility. You'll definitely find us. And we are on our website, nscf.org.za, for any information related to natural science collections. You're also welcome to leave your comments or suggestions or anything, even questions, on the Facebook page or the YouTube channel. Just a reminder, we are live on Facebook, and if you would like to talk to us, you can talk to us from there. Thank you everyone for making time to be with us this morning amongst load shedding and all that. And just to say, our thoughts and prayers are with those in the KZN that have been affected by the floods. And we hope everything will get back to normal Even if it doesn't, we hope everything gets better. So our thoughts are with you. Other than that, that's it for today.